This video is just a short introduction to the idea of how things that happen in inheritance that we've talked about already can in fact lead to variation. Now I'm going to be talking about a few different things. The first of those is a particular type of interaction between one gene and another. Now we've looked at this already in terms of alleles. We've looked at the way in which alleles interact. So for example, if you have a dominant and a recessive allele, they interact, and the dominant allele is the allele that is shown in the phenotype. We've looked at it with codominance as well. You have two different alleles, and the two different alleles both interact to create an intermediate colour, for example. So we have already looked at some aspects of interaction. What I want to do now is to start looking at how interactions can happen between different genes, not just the alleles of one gene, but different genes. Sometimes the genes are on the same chromosome but on different loci. So for example, here is a chromosome, here is the locus, and here is another locus. And you have two different genes on that locus. In this particular thing, you've got one allele, let's call it A, and another allele, call it B. And the point that I'm making is that whatever characteristic is dictated by allele A, the phenotype that controls, it only works if this particular allele is also there. So there's an interaction. So this is the dominant allele, so you'd expect it to show the dominant phenotype. But it will only do so if this other allele is also present. Sometimes it can occur on different chromosomes. So you've got two totally different chromosomes. There's the locus of one, there's the locus of another. Here is an allele. That's the dominant allele, so you would expect it to show the dominant characteristic. There's an interaction between that gene and one on this smaller chromosome. If the dominant allele is on this chromosome, then whatever characteristic this is showing doesn't in fact happen. It only shows the dominant characteristic if there's another allele of a different chromosome a different gene on a different chromosome. So this interaction between the two. So sometimes interaction can be between genes that are on the same chromosome but on different loci, different locations on the same chromosome. And sometimes the interaction can be between two different genes, totally different genes, on two different chromosomes. So that's the first type of interaction. Another way that genes can interact is if they are linked. And by linkage, I mean that there are two different genes occurring on the same chromosome. So in this case, two different genes on the same chromosome. This chromosome gives the dominant alleles, and the homologous pair to the chromosome, inherited from the other parent, give the recessive alleles, just as an example. Now in meiosis, you know that the chromosomes, the chromatids, move apart. And so you would expect this whole chromosome to be ending up in one gamete, and this chromosome would end up in a different gamete. But because these two genes are on the same chromosome, you expect them to be linked. So these two alleles will occur in the gamete. So here's the gamete that's formed, and you'll end up with allele A and allele B in that gamete. Here is the other gamete, and you have allele A and allele B in that gamete. So the two different genes are linked because they appear on the same chromosome. Now we come to a slight problem with that. When we looked at the study of meiosis, one of the things that happen in meiosis is something called crossing over. That process. Non-sister chromatids cross over and exchange genetic material. Just as a reminder, if I draw them in two different colours. So this crossing over happens there, and the result of that is that after meiosis is finished, they've exchanged some genetic material. So this chromosome is now no longer quite the same as it was. It's got the same genes on it, but not necessarily the same alleles. So let's say, for example, this initial red one had all dominant alleles on it, and this one had all recessive alleles on it. Now this chromosome has got all these dominant alleles plus some recessive ones, and this one's got all these recessive alleles plus some dominant ones. Now, if this happens, it's possible that genes that are linked get separated when it comes to producing the gametes. So if I put on my original diagram here two different genes, let's say there's a dominant gene there and the recessive gene there, and a different gene, the recessive gene here and the dominant gene here. So this particular chromosome has two dominant alleles, the dominant allele of gene A and the dominant allele of gene B. This chromosome has the recessive allele of gene A and the recessive allele of gene B. After crossing over, so before crossing over, let's wind back a bit, before crossing over, the alleles, the genes are linked. A is linked to B because it appears on the same chromosome. So the chances are that they will stay linked. If, however, crossing over happens between the two, which is done here, the crossover point is between allele A and allele B there, and allele A and allele B there. That unlinks those alleles. Now we've got capital A there, and little b there. We've got little a there, and capital B there. So, whereas the genes were linked before, after crossing over, it's possible that they are separated. I think you'll understand, because crossing over is random and can occur anywhere along the chromosome, and it can occur more than once. You can crisscross. So, for example, this line might cross that one and you go back again. So you've got two lots of crossing over happening. I'm sure you can understand that the closer together the two alleles are on the chromosome in the first place, the less chance they have of being separated by crossing over. Just to demonstrate that, here are two alleles very close together. Crossing over can happen anywhere down the chromosome. So here's the homologous pair, and here is the recessive allele of each of these two genes. Crossing over can occur anywhere down the whole length of the chromosome. And because there's a lot more of the chromosome on either side of these two genes than there is in the middle between them, the chances of the crossing over happening here are quite small. It could happen here, 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 anywhere up there. So the chances are that if they're really close together, they will remain linked and will transfer to the gametes linked. If they're further apart, like that, then there's obviously a greater chance that the crossing over will happen somewhere in this gap here, and that ultimately these alleles will become separated. And we can work out something called the crossover value. And you'll see that when you read the notes. But the crossover value gives an indication of the percentage chance of these alleles being separated. So if they're really close together, the crossover value is very low because there's a very small chance of them separating. If they are very far apart, the crossover value is high because there's a much greater chance of them separating. Now, to finish off, this is just the first of 3 different explanations for variation in organisms. You'll remember when we looked at meiosis, the whole point of meiosis is to create variation, and it does it in 3 ways. This is one of the ways that obviously, if this is the way that the alleles were inherited from your father, and this is the way that the alleles were inherited from your mother, here is your homologous chromosome pair. When this cell undergoes meiosis, if crossing over happens, and you exchange the alleles, then you've created variation. So whereas you might have had, let's say for example, you've got blue eyes and brown hair, because you've inherited a recessive allele for the brown hair, you'll still have, your children could possibly have blue eyes, but they'll have different colour hair. So crossing over creates variation. The second thing that creates variation during the process of meiosis is something called independent assortment. You remember that when the chromosomes line up in metaphase, they line up on the equator. Here I'm drawing 4 chromosomes. Here's the homologous pair of those chromosomes. Remember, initially you would have inherited 1 set of chromosomes from your father and the homologous pair to that chromosome from your mother. Here they all are lining up on the centre of the cell, on the equator of the cell, during the process of meiosis, metaphase 1 of meiosis. Now you know what's going to happen in this case. They're going to separate like that. And so all these chromosomes are going to end up in one allele, and all these chromosomes are going to end up in the other allele. But independent assortment means the way in which these things line up on the equator. There's nothing to suggest that they line up so that all the chromosomes that were inherited from your mother are on one side, and that all the chromosomes inherited from your father are on the other, they can quite easily switch around. So they don't line up like that, they line up instead like that. And perhaps the top one's done the same. Like so. So independent assortment means that each chromosome pair can be one way around or the other when they line up on the equator. And that, of course, is going to create variation in the gametes. Because now, this gamete is going to have these two chromosomes that originated from your father, and these two chromosomes that originated from your mother. Still the same allele, still the same genes, remember, but they could easily be different combinations of alleles. And this side here those 4 chromosomes are going to end up in the same gamete. So I'm sure you can see that if you're not just talking about 4 chromosomes, but you're talking about 22 pairs of homologous chromosomes, each one of which can either be one way around or the other, there are literally thousands of different combinations of the ways in which the maternal chromosomes and the paternal chromosomes can separate in the gametes. So that is a second way that variation can occur during meiosis. And then finally, the third way is during the process of random fertilization. Here are the chromosomes in a gamete. Here are the chromosomes in another gamete. If these gametes are, for example, sperm, and you had thousands of different ways that these could be switched around with the 22 homologous chromosome pairs, that follows that there are thousands of different ways that you can end up with the chromosomes in the gametes. So every sperm is different. And so it's totally by chance whether this gamete or this gamete fertilizes an egg. And so therefore, obviously, if this gamete fertilizes the egg, the person that ends up being produced from that fertilization process is going to be different from that one, because this one has got different alleles here, different alleles here, different alleles here, different alleles here. So random fertilization is the third way that variation can occur. So just to recap, we've got 3 different things. Crossing over actually allows chromosomes to break and rejoin on the other half of the homologous pair, and it can happen anywhere up the length of the chromosome. So any set of alleles can be mixed up. Secondly, the independent assortment, the way in which they line up in metaphase. Every chromosome, homologous chromosome pair can either be one way around or the other way around. And so there are thousands of possibilities as to the way in which they are lined up. And then ultimately, that means that you have many, many, many different types of gamete, all containing the same genes, but millions of different combinations of alleles. And so any one of these millions of different possibilities could fertilize an egg, which of course again is produced in exactly the same way. So an egg and a sperm have equally large numbers of ways in which the alleles can appear. So that is the idea of random fertilization.